In this video, we're going to talk about glycolysis. I know this looks like a lot, but I'm going to erase it all and walk you through it step by step, draw out each of the steps, each of the enzymes, and highlight the key enzymes and key steps that you need to know for test day. Before we get started, if you enjoy my content, be sure to like and subscribe to stay up to date with all of my content, and be sure to share these videos with your friends who are also struggling with biochemistry. It'll help them out, and it'll also help the channel grow, and I'd really appreciate it. All right, let's dive right in. I know what you might be thinking. If we're going to talk about glycolysis, why did I go ahead and draw a plasma membrane? Well, because I want to introduce the individual steps of glycolysis as problems that need to be solved. And then I'm going to talk about how the cell solves these problems. That way you have an understanding of the underlying mechanisms behind the individual steps, and you don't have to rely too much on memorization. So problem number one, glycolysis takes place in the cytoplasm, in the intracellular space. But when you have glucose in the cytoplasm, it can easily escape out of the cell through these fancy membrane proteins. So the first problem we need to solve is to trap glucose inside the cell, and this is how we do that. The cell solves this problem by attaching this negative charge onto glucose. That way it can no longer freely exit the cell and it's trapped inside. How does the cell do this? Well, by way of a kinase, because recall kinases will attach phosphates to molecules, and phosphates are negatively charged. So in this case, hexokinase will phosphorylate glucose to give you glucose 6-phosphate. All this means is that now glucose on its 6th carbon has a phosphate attached, and it can no longer exit the cell. And one important thing about hexokinase, I'm going to put a red box around it because it's really high yield. It's an irreversible enzyme, and the relevance of irreversible enzymes is that, and you'll see this when we talk about gluconeogenesis, but essentially because gluconeogenesis is kind of the reverse of glycolysis, the reverse pathway of glycolysis, but there are a few exceptions because some steps, such as this one, are irreversible. So you'll need a separate enzyme in gluconeogenesis to bypass this. All right, let's move on to the next step. The enzyme involved in this step is not very high yield. So you're going from glucose 6-phosphate to fructose 6-phosphate. These are isomers of one another, meaning they have the same chemical formula. So we're just rearranging the bonds over there. And the purpose of this step is to be able to allow the next enzyme to carry out its function, because the next enzyme, as we'll see, will phosphorylate fructose 6-phosphate as such. Okay, so PFK1 or phosphofructokinase 1 will phosphorylate fructose 6-phosphate at the 1' carbon to give you fructose 1,6-bisphosphate. All this means is that you have a fructose with 2 phosphate groups attached to it. Now I have 2 boxes around this one. The red one, recall, signified that this is an irreversible step of glycolysis, and the black one signifies that this is the rate-limiting step of glycolysis, the rate-limiting enzyme, which means that it's what determines whether glycolysis is going to run or not. So we need to be able to regulate glycolysis because sometimes we don't want to break down glucose. Well, the control happens over here. So before we move on, let's talk about what are the things that are going to regulate, inhibit, and activate this enzyme. All right, so I know this looks like a lot, but it's actually very straightforward. The things that are going to activate glycolysis are things that are going to indicate that the cell is energy deprived, right? Because the end result of glycolysis is to be able to generate ATP. So if you have high levels of AMP or ADP, that signifies that you have low energy. So you want glycolysis to be ramped up. On the other hand, if you have high levels of ATP, this signifies that the cell is energetically satisfied, so you inhibit glycolysis. Likewise, if you have citrate, high levels of citrate, it also indicates that the cell is energetically satisfied, because recall citrate is an intermediate of the Krebs cycle. So that means the Krebs cycle is running, and therefore you do not need glycolysis to run any further. Now, what's this part over here, PFK2? So the idea here is that fructose 6-phosphate can be phosphorylated by a different enzyme known as PFK2. It'll phosphorylate it at the 2' carbon, so you end up with fructose 2,6-bisphosphate. Turns out that this molecule will also stimulate PFK1 and ramp up glycolysis. Now, why is this important? Because this enzyme is activated by insulin. So that means insulin also activates glycolysis indirectly through this enzyme over here. And just as a side point, this molecule over here also shuts down gluconeogenesis. And this makes a lot of sense. Because gluconeogenesis and glycolysis are the reverse processes of one another, there's no sense of having them run at the same time. So this molecule, fructose 2,6-bisphosphate, when present, will activate glycolysis, shut down gluconeogenesis, and vice versa. Next step. In this next step over here, we want to take this 6-carbon molecule with 2 phosphate groups on it and break it down into 2 3-carbon molecules, each one possessing a phosphate group. As you can see, we have dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, these will interconvert between one another. So you'll convert from DHAP to glyceraldehyde 3-phosphate. So as you'll see, every step after this point is going to happen through glyceraldehyde 3-phosphate, but it happens twice, and that's because dihydroxyacetone phosphate is converting to G3P. So in reality, it's as if we have two G3P molecules. And this step, by the way, is the final step of the energy investment phase. What do I mean by this? The energy investment phase is where you invest ATP molecules in glycolysis. So thus far, we've only really wasted ATP molecules. As you can see right here, converting glucose to glucose 6-phosphate required 1 ATP, and converting fructose 6-phosphate to fructose 1,6-bisphosphate required another ATP. So thus far, it seems that we are at a net negative of ATP molecules, but it all pay off in the following steps, part of the energy payoff phase. So let's start with the first step. As you can see right here, the first step of the energy payoff phase is converting glyceraldehyde 3-phosphate to 1,3-BPG, short for 1,3-bisphosphoglycerate. Now, before we discuss this step, notice that this ends in the suffix ate, glycerate, which means it's a carboxylic acid. And this is obviously an aldehyde, hence the name glyceraldehyde. In organic chemistry, what does it mean when we go from an aldehyde to a carboxylic acid? Well, that is oxidation. We've oxidized the aldehyde to a carboxylic acid. And when you oxidize something, there must be an oxidizing agent. Well, that oxidizing agent is going to be NAD+, and that's why you'll see over here we've generated NADH. And I want you to carry this over with you for all of metabolism, whether it's fatty acid metabolism, the Krebs cycle, pyruvate dehydrogenase complex. The goal is always to reduce these high energy electron carriers so that you can take them to the electron transport chain and generate ATP. And in the same vein, while we're reducing these molecules, we are oxidizing the carbon skeleton. The more you can oxidize the carbon skeleton, the more energy you're getting out of it. All right, next step. The next step involves the conversion of 1,3-BPG to 3-PG, or 3-phosphoglycerate. Notice we've lost one of the phosphate groups. So here we had 2 phosphate groups, here we have 1. What happened to that phosphate group? Well, we combined it with ADP to form our first ATP molecule. And I forgot to mention here, notice how over here we went from a molecule that has 1 phosphate group to one that has 2. You might say, did we waste another ATP molecule over there? Well, no, we didn't. We didn't waste any energy because we just used inorganic phosphate over here. And if you're wondering why I'm not mentioning like the enzymes over here, because I don't think they're entirely high yield, but a lot of times you can just get it from the context. So here it would be glyceraldehyde 3-phosphate dehydrogenase. Over here it'll be 3-phosphoglycerate kinase. But again, they're not as high yield as these rate limiting enzymes or the irreversible enzymes. So the next step over here, the following 2 steps are actually not very high yield. So the first one, you're just rearranging the bonds. You're converting from 3-phosphoglycerate to 2-phosphoglycerate. And then the step following that, you're forming this molecule called PEP, or phosphoenol pyruvate. This molecule stores a lot of energy, and that makes sense because the next step, we're going to generate ATP, as we'll see. All right, so this is the final step. It allows us to generate pyruvate and ATP. It's carried out by the enzyme pyruvate kinase. And as you can see, it has a red box around it, and that signifies that it is an irreversible enzyme of glycolysis. So in gluconeogenesis, it would have to be bypassed by some other enzyme. Now, let's take a look at the big picture over here for this energy payoff phase. It looks like we've gotten 1 ATP over here, another ATP over here. So it looks like we've actually only got 2 ATPs from the second phase. Well, what's the point of glycolysis then if in the energy investment phase we deposited 2 ATPs and the energy payoff we've collected 2? That's a net of 0. Well, that's actually not the case, because recall that this energy payoff phase happens 2 times, and that's because the HAP will convert into G3P, so all of these steps will occur twice. So that means we'll get 2 ATPs, 2 NADHs over there, 2 ATPs over here, and 2 ATPs down here. So that's a total of 4 ATPs, in addition to the ATPs that will eventually generate from NADH once it goes into the electron transport chain. So in that way, glycolysis makes sense from an energy standpoint. We're generating a net of 2 ATPs, so 4 minus 2, and of course, plus the ones we get from the electron transport chain. One thing I didn't mention was the fact that fructose 1,6-bisphosphate actually activates pyruvate kinase. So we call this feed-forward activation. This is very rare to occur in metabolism, so it's something that shows up on the MCAT. All right, if you found this video helpful, be sure to like and subscribe to stay up to date with all of my content.